Hi everyone, here we are again. Hello and welcome to another Saturday live stream, Saturday Microscopy live stream. Okay, I think we're going to get started now. Like every time, I would like to kindly request if you can quickly tell me if everything works fine, if you are able to hear me properly. And if this is so, then yeah, I think great. Then we can get started. Yeah, hello and welcome again. Usually, if many of you probably have already joined the live stream before, the first 5 minutes. I'm usually not starting right away because more and more people will be joining during the first few minutes. And it has become tradition that if you want to, that you tell me or tell us a little bit from where you are. And we've already got the first comments even before I started the live stream. So hello from New Zealand, from Honolulu in Hawaii, from Lebanon, yeah, and Vietnam, from Vienna in Austria. Okay, wow, great. From the Netherlands, okay, Scotland, Vietnam, Slovakia, Netherlands again, okay, United States, Dominican Republic, from Ireland, Bulgaria, I love this. Okay, so yeah, hello all Canada, hello all around the world. Today I would like to yeah, talk a little bit about the making of a permanently mounted microscope slides. You can see already over here, I've got a few mounting media here that I would like to show you. I've also got some slides that I already made before. Some of them are yeah, a couple of years old already. And I've got some yeah, samples up here. These are stained yeast cells that I would also like to permanently mount. And I also would like to show you at the beginning right now, before we dive in, a few pictures that some of you sent me, and I would like to share them as well. Yep, more and more people joining in from Romania. Okay, yeah, so Indiana, United States, New Jersey, United States. Okay, yep. Do you remember me from the last live stream? Yeah, I got so many viewers here. Yeah, names all sound familiar. Okay, so what I would like to do first is, for a couple of minutes, I would like to simply share with you a little bit off topic, I admit. Just share with you a few pictures that some of my viewers yeah, sent me. If you remember a couple of weeks ago, if you joined in, some people sent some pictures of the home lab. And I would like to do this as well because I received another picture of a home lab, and look, it's me in the background on the monitor, and something quite interesting here. Yeah, and the green microscope that you see over there, I was told, yeah, viewer already had it was the first microscope. I remember one of my first microscopes was also quite similar here. And over here, this one is actually quite interesting here, right next. Yeah, this one over here. Do you see this? Okay, this is a USB microscope, which is mounted on what appears to be. I mean, I don't know. It could be. I've got a similar one. It's a microphone stand, but it's also quite interesting. Yeah, to mount a microscope this way. Yeah, so. Quite nice. It's I always generally recommend that you reserve somewhere a little place for microscopy, because then it's actually much easier to quickly start doing a little bit of microscopy without having to carry around the microscopes. Yeah, and I also got already a few weeks ago, and I have to admit I have to apologize. I overlooked it in my flood of email. I got 2 pictures here as well, pretty nice ones, and yeah. The first one, actually wrote this down. The first picture, that's a European holly, which seems to be the cross-section of the stem of a European holly. And the other one, also very nice. Plants are very beautiful here in dark field. It's a plant called Wollemia nobilis in dark field. Look at all of those beautiful colors. Yep. I would also, yeah, invite you, if you also want to share a few pictures, so you can send them to me. Yeah, these pictures were sent from Marcel from the Netherlands. Thank you very much. Okay, so yeah, Indiana, United States, New Jersey, okay, Atlanta, United States, hello back. Okay, so what I would like to do today is the following. Before I start off. Yeah, here we go again. I would like to simply give you a little bit of a theoretical introduction into the mounting of permanently mounted slides. Yeah, there is. It's not difficult to make permanently mounted slides, but the choice of mounting medium is a little bit important. It depends a little bit on the specimen and also on your goals. With goals, I mean is how permanent do you want the slide to be? Because sometimes it's fine if you make a slide and it's you're able to store it for a few years. But if you really make them properly and if you spend a little bit of time also in specimen preparation, I read somewhere that some of the permanently mounted slides, they can survive a century. And indeed, there are some permanently mounted slides out there that were made back in the day, Victorian times. And these slides are now collector's items. Victorian times, 19th century, 18th, 19th century, around that time. Okay, so what I would like to do first is I would like to simply give you now a short introduction of the mounting media that I have over here and some of the advantages and disadvantages. I have to also tell you that not all mounting media are easy to obtain. You have to sometimes order them directly from chemical companies which are supplying laboratories. Sometimes not cheap. You can, in most cases, not simply order them over Amazon or over other stores. You might try to contact some educational supplies companies, but generally, I would say if you want to buy mounting media, at least the specialized one, I'm going to show you some cheap, easily available alternatives as well. You probably have to contact chemical companies, and then it's a little bit also a question of luck whether they're able or willing to send them to you over to private people and not to labs. Okay. Comments already. I would like to have an 18th century permanent slide. I would say probably 19th century is more realistic. And some of those slides are quite expensive. They're collector's items. So I would like, and I'm also later going to show you a little bit of how I made those paper covered slides. A few years ago, I was experimenting with paper wrapping the slides as well. And I'm also going to show you how I've done that. Why paper wrapping? Because many of the antique slides are also paper wrapped, and I simply wanted to keep up the tradition a little bit, and I also experimented around. The results were not perfect, I would say, but they were okay. So let's have a look here at some of the mounting media what did I have over here? But maybe there are a few of you who are completely new to microscopy, so I would like to maybe for those of you who don't know what a permanently mounted slide is, I'm just going to quickly explain it. Microscope slides here, they have on the slide there is a cover glass, of course, and in many cases what you do is you take a water sample, you put a water sample with some specimen on it, and then you put a cover glass on top. But those slides you cannot store for a long time because the water is going to evaporate, of course. But sometimes you would like to store the slides for a very long time. Okay, and if you want to do that, then the specimen has to be properly dehydrated. This means it has to be completely free of, or almost completely free of water. I say almost completely free because it depends on the mounting medium. But after it has dried, there is very little water there, and because of the lack of water, it also is able to store for a very long time. So somehow you have to process the specimen in a way that there is very little water, if any water at all. And different mounting media are suitable therefore for different purposes. And I've got a few of those here that I would like to talk about. So let's clear this away a little bit. And one of the first mounting media that I used is called UKIT. This is a mounting medium where you have to be a little bit careful with, because it uses a solvent, xylene, which is not healthy. So you have to use this mounting medium in a very well ventilated environment. If properly used, then it will give a very good and very well, a slide that stores for a very long time, provided everything is properly done. UKIT was the first mounting medium that I used, and as a matter of fact, if you, I'll grab my microscope slide box over here. This goes back for a very long time. If you just look up over here, when did I make those? I made them back in 1998. I used at that time, I used UKIT, and the slides are still fine. Okay, so this is an advantage of UKIT, is that it preserves the specimens quite well. However, the disadvantage of UKIT is that the specimen must be completely dehydrated. If there's only a little bit of moisture there, what's going to happen is that the UKIT is going to dry, and the specimen is going to stay moist, and maybe it's going to decompose. And then, when decomposition starts to take place, then this is a problem, because there are decomposition gases forming, and this might actually form bubbles over the time. But otherwise, UKIT is also widely used when making commercially prepared slides, and is also used in research and also in hospitals when they are preparing tissue samples. Even with automatic slide making machines, it's useful, and it dries fairly quickly. So after a couple of days it's already solid. UKIT is therefore a very nice mounting medium to use, but not one that I generally recommend for home use, because of the solvent which is in there, and because the specimen has to be completely dry. So one of my favorite mounting media that I used for many years is Uprool. And Uprool is a mounting medium which is used very extensively by people who are insect specialists, so to say, entomologists. So when you mount insects, for example, then Uprool is the mounting medium of choice. The reason is that Uprool allows you to mount specimens that contain a little bit of water. Okay, it's a little bit more moisture tolerant, and I'm able to dilute it down with alcohol. So if I, for example, have an insect in alcohol, then I can transfer it directly on a slide, and I can use Uprool, and it will basically then mount the insect. And Uprool has the advantage that even if the insect is relatively dark, the exoskeleton, the outside of the insects are sometimes very dark. Over the course of several days or weeks, it will brighten the insect, so it becomes more and more transparent. So in that sense, it's apparently the mounting medium of choice. And as a matter of fact, I bought this mounting medium from a store that sells equipment and laboratory equipment for people who are studying insects. So in that sense, they of course have it. And I just saw, it expired 10 years ago, 2013 it expired, but it's still fine. Okay, so I've been using it, and it's still okay. So then another mounting medium which might surprise you a little bit, but indeed it is also used, and that is clear nail polish. Clear nail polish. Clear nail polish has a slightly the disadvantage that it likes to shrink a lot. So when it evaporates, then it really starts to shrink, and sometimes bubbles form. And the clear nail polish, however, I've got I know someone who's been using actually a clear nail polish in the university laboratory. So for them, this was the mounting medium of choice. Now you're gonna say, why would anyone use something like a clear nail polish? Well, the choice of mounting medium not only depends on how compatible it is with a specimen, but it also depends on the refractive index. And for certain specimens, it has just the right refractive index, and it works quite well. So I've been also experimenting with nail polish, and here I recommend that you put the specimen. It's got to be completely dry, obviously, because nail polish does not like to mix with water. And what you do is you take you put the specimen into acetone or some nail polish remover, and then when you transfer the specimen from the nail polish remover into the nail polish on the slide, then it will be also fine. Okay, so and I've got the last mounting medium here, but before I talk about the last mounting medium, I'll go back always to the comments. Okay, biology is always expensive here. Well, sometimes we have to look for cheaper alternatives. And hello from South Texas. I'm new to Microscopy. Also here, I'm actually just about to purchase the Motic. Yep. So any info on making slides is super appreciated. Okay. So So you can't drink it anymore. It's past its expiration date. Ha, yeah, sure. No, you should not be drinking, obviously, and also try to avoid skin contact. The thing is that those mounting media, many of them contain solvents, alcohol, in some cases, sometimes other solvents, and therefore they do not turn bad. Okay, so therefore they do not spoil in the sense. But if you keep it longer in a bottle, it can actually, some of the solvent might escape a little bit and might become more and more solid. Okay, so what I have here now is that is my newest experiment, and it's called Elmer's PVA. Polyvinyl acetate, polyvinyl alcohol is basically a glue, and yes, there are commercial PVA mounting media available, but I found a very cheap source. Some of you might already remember this from my previous live streams. Yeah, that is the glue here, and it is clear glue, and I diluted it down with a little bit of water, and it works surprisingly well. And it has one significant advantage, the PVA glue: it is water-based. So in that sense, it is safe. Yeah, as a matter of fact, it even says here that it's child-friendly. Yeah, so there are no organic solvents that we have to worry about. And the other advantage is that I'm able to take organisms, water organisms, and I'm going to show you one, over here, algae. Yeah, now over here, it's a water organism, and I was able to mount the algae directly in the PVA glue. Because if I were to mount certain water organisms like algae and other water organisms in a mounting medium that contains a solvent like Uprool, for example, well, I cannot do that because the water and the mounting medium don't mix. And if you put it into alcohol, what's going to happen is that all of the pigments of the algae, the chlorophyll and so on, it's going to be washed out. Yeah? But because it's water-based, it's not a problem. Okay, so I think this is indeed something, if you want to experiment around making permanent slides, and you don't want to spend too much money, and you want to have a safe mounting medium, then I highly recommend that you grab yourself somehow a bottle of this glue here. The company also has white glue, but this one is the clear version, and then you use that and you dilute it down with a little bit of water, and you mix it, and then you let it stand for a few days so that the bubbles disappear after you mix it with water. Then you can use that. So I'll be experimenting with this here as well. Okay. Wow, hello from South Texas. Okay, so Texas, USA. Okay, 7:45 a.m. Okay, in Australia. Oh my gosh, and people are actually also joining in at the middle of the night at 3:45 a.m. Hi, I have a question isn't related with the slides. Is it worth to buy an objective from Olympus to replace a normal plan achromatic one? That's too difficult to say. It depends a little bit on the objective itself. Okay, and the standards have to be compatible. Okay, can you use a stain in the PVA for pollen slides? If you want to make permanently mounted slides in pollen, then there is a mounting medium that's called glycerin gelatin, according to Kisser. Glycerol jelly or glycerin gelatin is a water-based mounting medium, and it is suitable for pollen. However, there is a variation of this, and it's according to Kisser. And if you get if you Google it online, glycerin gelatin according to Kisser, then this is the standard medium to be used for pollen. I don't have it here, but simply because there was a question here. Okay, I bought 120 prepared slides from Amazon. It's very good for its price. Yes, that's another thing. Sometimes in a microscopy community, there is sometimes a little bit the discussion going on is it even worth making your own permanent slides, or should you not just buy finished permanent slides? And my answer is, well, it depends. There are certain specimens where you can make permanent slides quite easily, and why not? It's fun, and you can make your own slides. I've been doing this, so why not? But then again, if you want to prepare tissues from animals, for example, okay, animal tissues, then it becomes very elaborate and sometimes a little bit complicated because there are many preparation steps. You have to embed it in paraffin, you have to use a microtome to slice it, and so on, proper staining. So it depends very much on the type of specimen. Sometimes it's very easy to make, like today, and sometimes it's quite elaborate. Yeah. How do I extract a microorganism from a dish and dry it up? I like to make a permanent nematode slide or something. Well, I got nematodes over there. You got to be aware of one thing. You don't need to extract it. All you do is you take a drop of water containing the microorganism, you put it on the slide, and you let the water evaporate. But be aware that especially water microorganisms, they have a high water content, and when you dry them, then they will shrink. Okay, so that is a general problem with water microorganisms, that they will change their shape during the preparation process, and for this reason, water microorganisms are best mounted in a water-based mounting medium, possibly glycerin gelatin, which does not completely dry up. Okay, hi from South Africa, from Ontario, Canada. So if the glue is water-based, we don't have to completely dry up the specimen. That is correct. Okay, and because I've been talking now for a very long time, I'm going to now show you this one slide here of the algae, which I made back in August 2022, and I used this PVA glue. And what I did is I simply took some algae from my little jar. I removed most of the water, so it was not floating in water. I used a little bit of tissue paper. It was moist, but it was not swimming in water. I added a drop of PVA glue, mixed it a little bit carefully, and let it sit now for, I don't know, a couple of months to dry. But actually it already dried out quite quickly. So, and let's have a look at this here. I need to turn on the microscope. Okay, and let's put the slide in here. And yep, here we are. So these are some filamentous algae. Okay, it's completely out of focus, of course. Yeah. Let's go up a little bit with the light intensity here. Let's go up with the magnification as well. Okay, and what I've been checking here is you see that the cells basically they have pretty much retained their shape, and none of the green color of the chlorophyll was actually washed out. Because if I were to mount this in a mounting medium which contains some kind of alcohol or solvent, then what's going to happen is that the colors of the chloroplasts, the green chloroplasts, are going to be washed out. Yeah? And the whole thing is also going to start shrinking, but this didn't happen here. Okay, because it was a water-based mounting medium. So I think that this PVA mounting medium, the PVA glue, is pretty nice. I like it a lot because it's not only cheap but also very uncomplicated. And me being, of course, also a teacher, a biology teacher in school, I'm interested in safe mounting media that the kids can experiment with. And of course, glue is a safe, especially this glue is a safe one here. Yeah. So there was again a question about microscope objectives. Okay. I would so very quickly, generally not a good idea to mix objectives because you lose the parfocality. So this means you have to, if you change objectives, you got to change all of them at the same time. Otherwise, you're going to lose focus when you change magnification. Hello from Portugal. How do I make a permanent crystal slides? Okay, yeah, that's also something that I'm going to talk about. Because crystals have a problem in that it depends a little bit which type of solvent you're using, okay? What about something like hairspray? Well, I don't think that hairspray, you need a little bit of liquid, okay? And hairspray does not give you sufficient liquid. So, what I'm going to, my first mounting medium was Canada Balsam when I was a teenager, 1958, with reduction in mobility. I've resumed, okay. Canada Balsam indeed is a mounting medium that was extremely common and gave very, very long lasting slides if properly made. Again, the problem with Canada Balsam, if still available, what I read is that the sample has to be completely dry, and you have to make sure that the sample was placed in xylene solvent before you could mount it in Canada Balsam. But Canada Balsam is one of those traditional mounting media that has a very long time storability. So what I'm going to do now is the following. I'm going to simply start making a few of these slides here, and I just want to show you what I've prepared here. This is already dry, and you see that there's some blue stuff on here, and these are stained yeast cells. And I just want to show you now what I have done. And I simply have basically over here, I already got a little bit of yeast solution. So what I've done is I took some water and I added a little bit of dry yeast. I don't even know where to put the packet now, and allowed it to stand here. And what I've done then is I added a little bit of methylene blue solution. So I'm just going to show you what I've done before in the afternoon today, a few hours ago. Because of the drying time, I simply wanted to stain the yeast cells, and I simply added a couple of drops of methylene blue solution, and that's okay. Now I stained my finger, and that's pretty much it. Okay, just a second. Need to get myself some tissue paper. Okay, to wipe the methylene blue. So yeah, and that's basically what I did, and then I stained it, and I placed using a pipette. Yeah, using a pipette, I placed some drops on the slides, and I allowed it to dry for an hour or so. And now I'm able to, I'm just going to put this here away because we don't need it. I'm just going to now make a permanent slide using this first slide over here. Okay. Before I do that, I will remove my other slides that I have here, and then later I'm going to show you how I made the paper wrapping. Well, actually, I got to be very honest with you that the process that I'm going to show you is pretty repetitive. It's always the same thing. But let's try it first. With I don't know, just because it's here the PVA glue. There are two types of cover glasses that I have. Which one you use is pretty much a question of taste. There are round ones over here. They're kind of dirty and a little bit more expensive, and I've got some square ones, and I'm just going to use the square ones. Okay. When you take them, I always recommend that you, difficult to grab. Okay, here we go. When you take them, I recommend that you still dry wipe them a little bit. I'm super well prepared again. Where did I put the piece of cloth for wiping? Just a second. Okay, where is it? Okay, I'm just gonna take some tissue paper here again. Okay. So I'm just because wiping is a little bit important because you want to remove grease and whatever residue there might be on it from the manufacturing process. Yeah. So and then you can actually see that it's reasonably clean. And what we now need to do is the following, and I recommend a little bit of experimentation here as how much mounting medium should you use, because every mounting medium, some more, some less, will shrink a little bit when it dries, because of course there is a solvent in there, and when the solvent evaporates, then there's less volume there. So this means it's a little bit of experimentation as well. Wow, that's a lot. Yeah, so this is the PVA glue that I diluted down with a little bit of water. Okay. What I usually do is the following: I put a drop of the mounting medium on top here. Sometimes I, for example, when mounting insect wings or slightly larger objects that are flat, I usually put a drop first, then I put the specimen, and then I put a second drop on top. Okay, that's a possibility. And then I simply take the cover glass, drop it on it, let the capillary action do the rest. Or what you might also want to do is you simply carefully arrange it and press down on top here. Okay, so and then you wait until it's dry. Now I'm going to show it to you anyway because we're all a little bit impatient, but you have to be careful. Okay, and I'm not going to look at it with a high magnification because some time ago I really learned the lesson the hard way. Do you actually see, I'm going to use my toothpick here, that here on the side some of the mounting medium kind of spilled out from beneath the cover glass. Okay, that's not a problem unless you're dipping the microscope objective into the mounting medium. Don't do that. Okay. Actually, did that once, and then it was actually UKIT that I used as a mounting medium, a solvent-based mounting medium. I dipped my 40 times microscope objective into the mounting medium on the side, and then I had to clean the objective with alcohol, and I was very, very nervous when this happened, because I was kind of worried that I ruined my objective, but everything was fine. With alcohol and some solvent, I could actually get it removed again. So this is how it looks like. I want to show you over here as well. This one over here, which I made a couple of months ago. Look how much spilled out here on the side. It turned completely hard. And what I tried to do, and it's possible, I'm not going to do that now, but don't wipe it away. Yeah, let it dry, and then use some kind of a if you want to, it's cosmetics. You can use some kind of a sharp knife to kind of scratch off the material that kind of spilled out. Yeah, I mean, it's not necessary really, but I mean, if you want to make it look nice, then this is a possibility. So these are basically now the yeast cells with the PVA glue, and I'm going to put it under the microscope. But I've seen a few more questions, which I will basically talk about in a minute. Seen a microscope. Okay, so I'm going to again always. I'm not going to go too high with the magnification. But the yeast cells are not very big. So that's now the 10 times. And here they are. They look like little dots, of course. Let's go up a little bit higher. Yeah, yeast cells. I don't know. Should I try 40 times? I'm a little bit nervous. Nah, I'm too nervous. I don't want to try 40 times. Okay, so because if I really dip it into the mounting medium, then but you get the idea. You see that there are nice blue clusters here, and I think it's gonna work fine. Okay, So this is basically the PVA glue, and I don't know. You know what I'm gonna do is I'm going to. Actually, I should label it now somehow, and I might actually wait until next week, and then next week we can actually have a look whether the cells actually still look the same way as they do now. Okay, so a couple of questions here. There is not much difference between okay, here again about thank you. Some other viewer. Brent was answering the question about the objectives here. Good evening from Hungary. Okay, I've actually first tried dark field microscopy after I saw you do self-filter video. Okay, yes, nice stuff. I need to make some permanent slides. Hello from Tennessee. Will the chlorophyll color fade? The thing is the following. This refers back to the slide with the algae that I have here. The question is, will it fade? I've got some very old commercially made permanent slides. Yes, the color did fade. Because I think, pretty much all pigments sooner or later will fade. And depending on the compatibility of the mounting medium with the pigments, and this of course can also influence this. I noticed that methylene blue appears to be your preference for staining. Okay. Did you try eosin Y? I said, no, it did not. Yeah, long time ago in university I tried it, eosin, but not recently. Okay. I try to use the 40 times magnification, but it crashes in the slide. Well, if you use the 40 times magnification properly, that means you first focus with a 10 times objective, and then you switch to the 40 times when it's in focus, then it should not crash. Okay, or the 40 times objective is from a different series, and therefore it has you lose part focality. This could also be a problem. Okay. Would diatoms mount in PVA? Why not? But I think PVA glue is not the medium of choice for diatoms, because here the refractive index is really important, because the diatom shells are transparent, and in order to see them properly you have to have the correct refractive index. Okay, I found few dead tardigrades in moss recently. Would the PVA method work, considering they already have bacteria around them starting to do their thing? Yeah, what's going to happen is the following. Of course, the bacteria decomposing, but as soon as it dries up, then bacteria will of course also stop working. because living things need water, and if something is dry, then it is preserved. How much should the PVA glue be diluted? Essentially, there is not a very. I did not measure it out really. Honestly, I've seen it's kind of thick. Yeah, so I added a little bit of water. Several milliliters, and then I thinned it down this way, and that's basically what I've done. Maybe I would say I've added maybe 20%, a fifth of the volume. Yeah, and again, it depends a little bit. The thickness depends a little bit on the specimen itself. If it's it should flow without problems. But I would say the advantage now is that I thinned it down to the extent that it does not make any strings, because the original glue, the one over here, okay, I can actually show this to you. I'm gonna use an old slide over here. If I were to use this here directly, just gonna show you. Okay, you see that it makes strings. I don't know if you're able to see this, yeah, which are a little bit annoying, and I consider this too difficult to use, and it doesn't flow properly, and then when you put a cover glass on top, it doesn't spread evenly, and for this reason I thinned it down a little bit, and I would say when it does not make any strings anymore, it doesn't pull strings, then I think it's properly diluted. Okay. Would butterfly scales mount okay and show color in PVA? Well. I don't have a butterfly scale now, but I do have in UKIT, I do have mosquito legs. Okay, and there are also scales on there which are similar to butterfly scales. So let's have a look at this, and then I'm going to mount another slide. Okay, so these are the yeast cells. I have to take it away and put it somewhere where I can dry it properly. And here, these are now mosquito legs mounted in UKIT. See, and somewhere it's a little bit. I think I thought I remembered that there were some scales visible here, but maybe not. Yeah. No, where are they? No. Yeah. Okay. Butterfly, not a butterfly. A mosquito leg. No, I don't see the scales here. Maybe they were in a different one. Let me see. Let me focus a little bit. Ah, no, over here. Here. Do you see this? This is one of the scales. These are scales of the mosquito leg, and butterfly scales. They look kind of, depending on the species, they kind of look a little bit similar. Yeah, but this one is not mounted in PVA, but this one is mounted in UKIT. Okay. So just to show you, and yeah. So, what total magnification does your microscope have? Well, if I use oil immersion, then 1,000 times. Okay. Does mounting work for all types of specimens? Yes and no. I mean, of course you can try to mount pretty much all type of specimens. The only problem is does the processing, the dehydration, the removal of water and so on, and the mounting itself, does it destroy the shape to the extent that the specimen is not useful anymore? And this can happen sometimes, happens quite often. Yeah? So I personally also think that water microorganisms like single-celled the paramecia and even tardigrades, I think they're more interesting to look at when they're moving around. Right, and when they're mounted in a mounting medium, well, then you just see them, but yeah, they don't move. Yeah? So, however, you can make, some people do make permanent slides of water microorganisms, single-celled water microorganisms after staining them, because they're using some staining technique for identification purposes. Yeah. How do you label them? Okay, that's also another thing. Very quickly, I go back here. How do I label those slides? I made those labels here. You won't believe it, in PowerPoint. Yeah. So what I've done is really, you can in PowerPoint you can draw boxes and you can specify the size, and then I simply typed it in there, and then I copy pasted it and printed it and cut it out using scissors. That's it. And I glued it on here just using regular glue. Yeah. So these are basically printouts that I made. I show you something else over here. The first slides that I made here, I hand labeled, and I got myself some stickers. These are just self-sticking stickers. Okay, maybe you can. I don't know if and simply glued them on here, and I used some ball pen to label it. Okay, so there are different ways how you can do that. Yeah? So is there any way to make homemade dye for the specimen? Homemade dye. Well, what you can try to do is you can try to use the ink of a fountain pen. Or some intensively some food colorings. So there are some dyes available that actually also work. Okay. Yeah, here we go. Fountain pen. Yeah, here. Yeah. Yes. Thank you very much. Okay. So would certain concentrate of etol help fix dehydrate samples before mounting onto the slide? Did not try that yet. You can try it. Please. I have not tried it, but if you want to try it out yourself, and if you want to write me an email because you're experimenting with certain techniques, then tell me and I will share the pieces of information. Okay, so let's move on a little bit. So this was basically the PVA, and now let's try another one over here. Okay, and I'm going to now mount this in Uprool. And pretty much the same story. I need to prepare myself first a cover glass. Look at all of those slides here. I just piled them up, not very nice looking. I should be a little bit more careful here, but I'm just going to, for the sake of convenience, push them to the side. So let me also clear this space a little bit. I don't have a lot of space here. Just got the camera mounted on top, and a microphone is here next to me. So so let's put this over here a little bit, and here let's try Uprool mounting medium. Uprool has a very typical yellow, see, not enough, yellow appearance. Okay, so that is a very, and it has a very nice smell, because I don't smell it, but yeah, it looks like, yeah, like, so you put a drop on here, and of course a cover glass goes on top, and something that is a little bit different of concerning Uprool is, well, of course it contains a solvent, so we're going to see how it reacts with the dye, with the methylene blue. And another thing is you have to now store it horizontally for about 6 weeks, because it takes the Uprool a long time to dry. This can also be an advantage, and the reason is because when you're mounting insects and other small animals, then the long drying time gives it enough time for the mounting medium to infiltrate and to go into the insect, and also to preserve it inside out. And this is actually an advantage if the mounting medium is slow drying. But yeah, I will actually also put it under the microscope now. Okay, so scene microscope again. You got to be careful with this, that you do not dip your objective into the mounting medium. Yeah, and yeah, you do not see the yellow color. That's also an important thing. Yeah, and this is how it looks like. Yeah, under the microscope. So I can go up a little bit more. Yeah, and refocus. Yeah, you see that looks pretty much the same. Okay. So, but again, the Uprool does have the advantage that it has a very strong clearing action when you're mounting insects with it. That's why it's the mounting medium of choice for people who mount insects. Yeah, so far these were actually easy. In case you don't know, these were actually quite easy specimens, because that's why I used the yeast, because it's all completely flat. Yeah, it's all completely flat, and therefore easy to mount. Okay, so let's put this away again. Yeah, in the meantime, I'm going to do a little, how do you say, sidetrack. I'm going to do a little excursion before I mount the others. And I would like to simply show you how I prepared those paper-covered slides. Why did I do that? Well, actually simply because I wanted to make paper-covered slides. There's no specific purpose or meaning behind those. Of course, you can always say, well, they're a little bit more, yeah, they don't break as easily and so on. But actually, I just wanted to try it out. So a few years ago, 10 years ago, I don't know, yeah, even, I made some of those slides, and I'm going to show you now a video. And as a matter of fact, if you check the channel and if you Google for slide wrapping, I actually showed it to you also in a video in either this channel or the main channel. But here is the video. Okay, so that's basically what I've done. I used some regular paper, and I using a hammer and this hole punch. I simply punched out a hole, and then I used a old credit card to apply some wood glue, some water-based wood glue. I'll just show you where's the punch. Here it is. Okay, yeah. There were different sizes, but I think there might be, I've seen some where there are actually normal hole punches that also have a diameter like this. Yeah, some kind of a machine where you put the paper in and then you press it down. Yeah, and I simply use this one over here. I think, yeah, and it kind of worked. Yeah, I used then I put the slide on top. Okay, carefully pressed down on it, and this is just regular wood glue. Yeah, and it makes the paper very soft at the beginning, but after it dries, it makes a very solid and strong connection. So I've folded over the edges, and I press down a little bit to make sure that the paper contacts the glass slide, and then I added some reinforcements for the corners, because I was kind of worried that maybe the corners are going to chip off. Yeah, I was just simply experimenting. And then I added another layer on top. And then I simply waited a day for it to dry. I also added some slide labels just to make it look nice. And if you look carefully over here, you can see that actually over the place where the specimen is, I put some newspaper. I covered it up. And the reason is because I tried something. I tried to spray paint it with some clear, how do you call it? Yeah, paint, not the video loops. And this basically I was kind of protecting it. And I think, it makes it a little bit shiny, the spray paint, and also a little bit more water resistant. But I think it's not necessary. Yeah, it was just a little bit of fun, and I just want to share it with you if you also want to experiment around. Yeah. So a few more comments. A lot of comments in the meantime again. Okay. Do you mind if you can zoom further using DSC to view the structure of the mosquito's wing? Okay, why not? Okay, you're asking. Mosquito leg, mosquito wing. Yeah, here I've got a mosquito wing. And I'm going to, this one is, I just showed to you here, mounted in UKIT back in 2021. Okay, so that's not the Uprool that I normally would have used, but this is the UKIT. And let's just have a look at the mosquito wing. And here, because it's completely dry, I have to find it. I can zoom in much more. Here we go. Okay, here that is a little bit out of focus. Here we see the scales better. Okay, a lot of dust and dirt. So this is now the 10 times. This is now 20 times. And I don't know, because don't forget one thing, that because of the mounting medium, I don't know if the DIC gives you the colors that you would expect. So this is now 40 times. So I think this is the filter, okay, and this is 40 times. And you see if I change the DIC prism, I do have a color change here, okay. And here you see some of the scales of the mosquito wing, okay. So this is let's go in all the way 60 times. I have to change the filter here again, give it a little more light. Condenser, and that's basically the mosquito wing at high magnification as you requested. Okay. If objective crashes into the slide, then look sideways at the stage to lower the objective while observing it 40 times. Okay. Yes, this is a comment concerning the problem of the crashing objective. ETOL, I did not is a mounting medium. I did not try that yet. Okay. Yeah, if the mount is too thick, that's also a problem. If you're actually using too much, if the distance is too high, the cover glass is too high up, then of course it's also able to crash. Okay. Have you tried to rehydrate a dried specimen? No, not tried that yet. Okay. I tried using potassium permanganate. That's a very pink, very pink, highly oxidizing substance. Okay. It works somehow. Okay, cool. Okay, it's a kind of a strong lie. Okay, I'm just quickly going over. There's conversations happening here. Some conversations in the chat that I'm just skipping over. I'm looking for questions. Okay. What is the brand of your Uprool? I've seen different manufacturers with very different formulations, and heard some do not hold well over time. Honestly, this one is from a German company. It's from also a school supplies company. No, it's not from it's a PVA Glue. Yeah, I don't even know if they're manufacturing it. Sometimes my suggestion, there is no brand on it. I simply bought it from a retailer, and I guess they basically bought it in large quantities, and they simply refilled it in bottles and then sent it off. Yeah. These days it's sometimes a little bit difficult even to get liquids which contain solvents to get them mailed for safety reasons. Yeah. So that might be another problem here. Okay. Hello from Mexico. Okay. Does the extra slide thickness from your video make it harder to use the 1000 with the oil objective? Okay. Does the extra slide thickness from your video make it harder to use the 1000 times mode with the oil objective? So I guess this means a little bit is using because of the mounting medium and maybe the paper and everything here. If it's using a lot of mounting medium, how easy or how difficult is it to use the oil immersion objective? And the answer is I never ever use oil immersion on permanently mounted slides. The reason is because it's very difficult to actually remove the oil again, and then there is some kind of an oil layer smear on it, and sometimes if the oil were to contact now the slide label, it's also not going to be very nice. So my general, that's basically the rule that I made for myself, is that I never use oil immersion. On permanently mounted slides. And indeed, it is correct that if you want oil immersion microscopy to work well, the specimen has to be extremely thin. That's correct. And indeed, sometimes the specimen, especially if you use too much mounting medium, is not thin enough. Okay. So this is kind of the thing that I just wanted to share with you over here. So let's move on a little bit. What time is it? Okay, well, 52 minutes. So we still have a little bit of time. So I tried the PVA glue. I tried Uprool mounting medium. Let me remove all of those bottles first. So let's now try clear nail polish. Clear nail polish has the advantage that it's of course very cheap, very easily obtainable. However, I found the main disadvantage is that it really likes to shrink a lot, and this means bubbles. Bubbles form during the drying process. And there is, however, one way, one thing that you can do to minimize this, and that is to maybe not allow it to dry first a little bit before you add the cover glass, because then it starts to shrink before you add the cover glass on top. Okay, so let me prepare as always how do you call this? Cover glass. How do you call this? We wipe it again. Okay. And I have to find a place to put it. I put it over here. Okay. So, and instead of simply using a pipette, I sometimes just use the brush itself to tap some of the. Yeah, and I see that this is already pretty thick. So normally what I would do now is add a few drops of acetone or nail polish remover. Okay, and yeah, it's very thick. It doesn't flow very well. That's normal because over time a little bit of the solvent will always evaporate. And cover glass goes on top. Just make sure it's only one. Yes. Okay. You can see it doesn't spread quite as quickly. So let's take again the tweezers. Tap a little bit to make sure that it spreads properly. Okay. And let's put it again under the microscope. Okay. Here we go. And here. And always back with the magnification. Let's take out the mosquito wing. And again the yeast. I mean, always the same. Of course, what did we expect? Yeah. So that's basically now in clear nail polish. So maybe we're gonna have we're gonna have a look at this next week after everything's dried, and then we can actually really investigate it a little bit. How much did it shrink, or did any of the stains, any of the methylene blue, did it get removed? We can try all of that. Okay, and Is the yeast heat fixed and then stained? No, that is also a good question that I would like to talk a little bit about. Thank you for the question. What is heat fixing? And the reason why it's not heat fixed is because of the way that I stained it. I will tell you briefly what normally you do in the microbiology lab when you're staining. What you do is you place a suspension of the yeast, of the bacteria on the microscope slide, and you allow it to air dry completely. And then you pull it with the specimen pointing away from the flame. You pull it through a gas burner very quickly 2 times, and then you test it. It should be hot enough that you're just able to it shouldn't hurt. It should be very hot, but it shouldn't burn you. And then they're heat fixed. And what happens during this heating is you're not burning the cells, but they're baked to the glass. It's a little bit like when you've got an egg in a frying pan, and when the egg starts to stick to the frying pan, because the proteins in the egg, they will make cross linkages to the frying pan, just like the proteins on the cell surface of the cells will make cross linkages to the microscope slide. And this is called heat fixing. And this allows you to take then the whole slide and put it into a staining solution without the cells being washed off. Okay, so that is the standard way of doing that. And what I have, however, done is I used a different way of staining. The easier way is I simply mixed the yeast with the methylene blue. I directly put it on here, and I simply allowed it to dry. Okay, so no, they are not heat fixed. They were just stained, and then you basically let it dry. So this basically the next thing is what's the next thing? The next one is UKIT. Okay, this is the one that actually where you actually should have some ventilation going on. And I filled it into this bottle here. And what I'm going to do now, okay, as always, of course, you want to prepare the this one over here first, the cover glass first. Okay. So, Elmer's PVA. Let me just remember then I had Uprool, then I had I'm going to label it later. Then I had the clear nail polish, and now I'm going to use, I'm going to now use UKIT. I dropped the microscope the cover glass here. So the problem that I have here now is that I don't have a pipette. Okay. Of course, I do not want to use a plastic pipette, because I'm kind of concerned that the plastic is going to be dissolved by the solvent in here, and because I don't want to use that, I'm going to just do the following. I'm going to put this in here. I'm going to do it a little bit the cheap way. I'm just going to use a toothpick and then allow it to drop on. This is also very thick. Okay, it's too thick, I think. Okay, so I'd have to add a little bit of solvent, xylene. solvent which I don't want to use. So this toothpick here I have to remove somewhere, so I'm going to put it into the trash can, and then later I'm going to throw it out because the solvent, I don't want to have too much of the solvent in my room. And let's put it now on here again. Okay, and as always, you see this one is not running very well. Okay, it's too viscous, so over the months, apparently it must have lost quite a bit of the solvent already. Yeah, and let's try the same thing, and let's put it again under the microscope. And yeah, what did you expect? same as before, right? But maybe next week, maybe we're able to see a little bit of a difference. Yeah. But I think you get the idea that so far all of the mounting media produce, at least right now, pretty much a very similar result. If all of a sudden some of the methylene blue were to disappear or to kind of spill out or be washed out, then this of course would mean that the staining and the mounting medium are not compatible with each other. Okay. Hello, is it possible to do a permanent slide with a squared cover slip? Yeah, well, that's what I've been doing right now. But I will also show you something a little bit with round cover slips. Okay. So just a second, let me put this away again here. So that is now a UKIT, okay, over here. So, yes, did you find a way to mount the Artemia sea monkey, if I remember correctly, disintegrated with the glue? Yes, it disintegrated. Last week or 2 weeks ago, I mounted a sea monkey using the glue, but I think because of when the glue is drying, there are certain tensions in the glue, and this kind of caused it to deform a little bit. So there are also some physical tensions that might be exerted on the specimen during the drying process. Yeah? Doesn't heat fixing change the specimen? Let's put it this way. Chemically the proteins denature, but essentially with bacteria it works, and with some other cells. Therefore heat fixing is actually a standard procedure when fixing bacteria for staining. Yeah. So this is actually very important. And we've got a comment here also in Spanish. Okay, so let's do now the following. I would like to show you now something else here, and that is the following, this one here. A couple of weeks ago, I showed you my so-called slide ringing table. And what I've done here is I've actually made a little ring using my slide ringing table. For those of you who don't know what it is, I'm going to give you a very short demonstration, okay? Because I would like to now make a so-called a dry mounted permanent slide. A dry mounted slide is one without a mounting medium, so it's basically in air. Has certain advantages and disadvantages again. Okay, so I'm going to show you how I prepared this here, and of course you need a slide. So I'm going to take a slide, and I've got this slide ringing table here. Where is it? Here's the slide, and it looks. Let me show you it. Yeah, it looks like this here from the side, and I can actually spin it here. Okay. So I put the slide here directly in the center. There's some clips over here, and just make sure that it's more or less in the middle, everything, and then I'm able to spin it. And what I'm going to do now is I'm going to put a ring. This is nail polish, yeah, which I made very thin by adding acetone, because it's got to flow well. And then you spin it, and then you kind of make it. You do this, okay? You allow it to dry a little bit, and then you add a second layer. That's all I've done. Okay, and then I basically, removed it. I allowed it to dry, And this is how I got this here. And in the middle, as you can see, I also added the yeast suspension. And now what I would like to do is the following. I would like to now add this round cover glass on top without a mounting medium in the center. And the ring is very important. Let me again dry wipe this here, the round cover glass. The ring is very important because otherwise any nail polish is otherwise going to touch the specimen, and I don't want that. So the ring kind of prevents that the mounting medium, or at least hope that it's going to prevent that the mounting medium actually starts to spill inwards. So the only problem that I have now is how am I supposed to do that? Because if I put it on here and if I put a ring around it, I might actually shift the cover glass. So what I'm going to try is I'm going to try to glue it down a little bit, using a little bit of glue on one side and on the other side. Okay, and just to place the cover glass, okay, so that the cover glass is kind of a little bit held in place. Okay, maybe it doesn't look quite as nice with those two dots here. Yeah, but that's simply something that I've been experimenting with. Or another thing that might actually work better is if you make the ring and you don't wait for the ring to completely dry. So if it's still a little bit sticky, okay, then maybe it's gonna work. Okay, so how am I gonna do this now? I'm going to put this over here. I have to recenter it again. Okay, completely. I'm going to do it here, and now I'm going to try to spin this, and I'm going to try to put some nail polish right on the edge here, and to kind of connect the cover glass with the slide. Okay, a little bit like this. A little bit the mounting medium, some of the nail polish will actually go beneath the cover glass. That's of course the whole point. But it's not going to spill directly all the way to the center. If you have, and this, by the way, because there was a question about preserving crystals on the slide. For example, if you have very thin crystals, you can sometimes not use a mounting medium. Because maybe the crystals will start to dissolve again. So for example, if you use make crystals using, I don't know, let's say salt or sugar in water, and then if you add a water-based mounting medium like the PVA glue, the crystals are going to dissolve again, right? So you have to actually use a mounting medium which does not dissolve the crystals. But sometimes maybe because of the similar refractive index between the mounting medium and the crystals, maybe the crystals don't look quite as nice. So sometimes it's nice if you somehow have some kind of protective covering on the crystals, but still everything in air. So this could be a possibility here. Okay, yeah. So this is now hopefully connected. I see that it is connected. Okay, I might want to let it dry a little bit and then maybe add another ring. And I'm just going to look at it now in air. Okay, so that is now the same yeast suspension in air. Yep, here is the, and you're probably going to see that it might not look quite as nice. Look at this, isn't this kind of surprising? It looks much darker. Now, why is that? Well, this is nothing unusual. Very often, air-mounted specimens look much darker, and this has to do also with the large difference in refractive index between the specimen and the surrounding air. Okay, so this is shows that actually adding a mounting medium actually makes the specimens appear to be more transparent, but they are not more transparent. It's simply the way that the light is refracted is different. Not as much light is refracted away, and therefore more light is able to go through, and therefore the specimens look brighter. And here you can see in an air-mounted slide, the yeast cells look much darker. Yeah, it has to do with the difference of the refractive index. Yeah, so this is basically one way how you can make slides like this, and I know that most of you are going to say, well, it's all very nice and good, but I don't have a slide ringing table. Yes, some people made them themselves, but if you don't have one, and most of you don't have one, and you also do not want to spend the time to make one yourself, I'm going to now show you a very, very cheap and easy way to make a somewhat improvised ear-mounted slide. Okay, and that works like this. The easiest way, of course, is to put a thin specimen on here and then simply use some sticky tape and tape the edges of the cover glass. I'm not going to show this to you because it's kind of self-explanatory, right? You take some tape, I'm just going to make it on one side maybe, okay? And you basically do the following. You make, you connect it here on one side, and then you kind of tape it over the specimen. You know what? I'll just use my reel. I've got over here, this one over here. Why not? Yeah. All you do is you take this. It's flat enough, and you take it over here on the side, and you kind of tape it across. And this is actually a very fast and easy way to protect the specimen. And you get the idea, right? Now, what happens, however, if you would like to basically use specimens that are a little bit thicker and you do not want to crush them? That could be another question, okay? And it should also be ear mounted, or maybe not ear mounted. Maybe you just want to make a permanent mount. Maybe it should be very thick, okay? So what do you do in this case? Yeah. Generally, don't forget one thing. If it is very thick, then the drying time is extremely long, okay? So you might take this into consideration. Now I dropped my pipette. Just a second, I gotta pick it up. Yep, this is what happened. Yeah, if it falls apart, you just put it back together again. Oh, okay, the other part also fell off. Yeah, okay, I'll just leave it there. I just wanted to show to you how this works. You can also make a spacer in a different way. All you do is you take again some nail polish. You put a drop of nail polish here on one side. Yeah, you use the nail polish as a glue to fix a cover glass here on one side. You use another drop of nail polish, let's say over here, to do another one over here, and now you have your spacer. And of course the third cover glass goes across on top, like this. Yeah, and you can also use you put a specimen in between here, and then what you can do is you can again put a drop over here, a drop over here, and you simply glue it across like this, okay? Also a possibility to, I know this, I've done this very quickly now, a possibility to preserve, for example, crystals in air, right? You have now a spacer here, which does not crush the specimen which is beneath it, and it's still surrounded by air. I've done this very quickly now simply to illustrate the concept. Okay. So when it comes to natural colors, I tried boiling red onion skin in water for 30 to 40 minutes and then straining the water. It can be used as a stain. Yes, basically the red onion skin, these are called anthocyans. These are natural colors of plants. If sufficiently concentrated, it might also work. Okay. Have you experimented with multiple stains to identify different locations in your samples? Not so much yet. Okay, well, I mean, I've got my standard stains like methylene blue. I use iodine staining. Okay, for example, I use the blue fountain pen ink. It depends a little bit of what I would like to see. Yeah. Hard drive platter mechanism also works as a slide ringing table. For crystals and other three-dimensional stuff, those glasses with a recess could work. Yes, there are some microscope slides out there which are a little bit thicker. And which have a so-called an indentation. They're concave. You can try those as well. However, what I found is that sometimes this concave indentation is not always completely smooth, and sometimes those slides are also a little bit more expensive. Yeah, but it's certainly possible as well. Okay. Instead of fixing the cover glass using the sticky tape, why not just use the sticky tape as a cover glass? We're gonna do that, okay? So, thank you for asking, and I will tell you why it works half well, okay? The problem is that the sticky tape is not very smooth, and therefore you can see the glue, and this is sometimes also a problem. Okay, so for example, let's use the following. Let's use the sticky tape as a cover glass. Okay, and when you put it under the microscope, then, by the way, I've been doing this already when I was collecting some face mites. Okay, then at a low magnification it might be okay, but at a higher magnification you can actually sometimes see streaks. Oh, actually it looks much better than I expected. But over there it is. Okay, those tiny fine lines that you see over there. Okay, that's basically the glue. I'm gonna use now the arrow to point to it. Do you see those lines here, the edges here? Yeah. So that's basically where the glue and the specimen where they interact. Okay. I'm just saying that this is sometimes a little bit of a disadvantage, because you're able to see the glue of the sticky tape. However, one thing that where tape is very good is, for example, by collecting dust specimens, or if you want to collect skin cells, or like maybe you've seen some of my earlier videos. Yeah, if you glued it over here and then collect some skin mites. Yeah, so that is actually quite well possible. Okay. Yeah. So yeah, centrifugal. Okay. Yeah. So there's again a centrifuge question. So what time is it? 1 hour 15. So is there anything else that I wanted to show you? Just let me see. Yeah, I wanted to quickly also show you maybe some of my earlier specimens here, and I wanted to also show you here some of the ones that I made a long time ago, and simply to show you after 25 years if it's still working or not, and how stable it is, because there are some of the specimens which are not quite as stable anymore. I would like to show this to you. Okay, so this one is the one that I made back in 1998. Wow, 25 years ago. It was one of my earlier slides, and it's a fly, parts of a fly. Oh, here it is. Okay, so you see over here a leg, a wing, and I don't know, another leg. I'm going to show you now a slight problem. What can happen if the specimen is not properly dehydrated? I'm going to show this to you here. Okay, I need to, of course, change over to the microscope view. Yeah, so far, yeah, that is a leg. It looks okay here, actually. Excuse me. Okay, but yeah, also okay over here. Looks quite okay. But what you see over here is on the side is there is a huge air bubble. Okay. Yeah, so over here, this is a huge air bubble. And I don't remember that this air bubble was actually there, because I remember that I was carefully making those slides. And sometimes what can happen over time is that if the specimen is not completely dry, then bacteria will start to decompose the specimen, and they will produce so-called decomposition gases, methane gas maybe, or CO2, and these can accumulate, and then sometimes bubbles are able to appear. So what my recommendation is, especially if you have thicker specimens, leave them in alcohol for several days and make sure that the alcohol is able to reach all parts of the specimen, because the alcohol will not only remove water, but it will also kill off and disinfect bacteria. Okay, and then if there are no bacteria around, then of course there will also be less decomposition going on. Yeah. So basically you see, okay, I need to flip this out. Yeah. So you see that I think that over here, this is where, yeah, with time the bubbles have started to appear. Yeah. So, but just wanted to show it to you. And let me go again back here. But otherwise, you see that the specimen, even after 25 years, looks kind of okay. I mean, these were one of my first specimens that I prepared. Yeah. And yeah, if it survived 25 years, and I think there is no reason why it shouldn't survive another 25 years. Yeah. Yeah. So there are some comments about obtaining natural colors from food. I think what's important is that the colors and stains, whatever source you use, are sufficiently highly concentrated. And then, of course, it depends with what they are able to react. So if the stain itself is more alkaline, like for example the methylene blue that I have is more alkaline, then it will react better with acidic parts of the cell. For example, DNA, which is an acid. So it will actually, the methylene blue is pretty good in staining DNA. Yeah, just wanted to show this to you here. A slide which is a little bit older. Yeah, older. I mean, 25 years at least, but it's still reasonably okay. Yeah, and yeah. What else do I have here? PVA glue. So there are some other some specimens that are less problematic, like the one over here is the fur of a cat. Yeah, which is already dry anyway. Yeah. So also mounted in Uprool, and let's see. Yeah, and there are no bubbles, and the cover glass still seems to stick quite well. Yeah. Yeah, I think the slide is a little bit dirty. That's why I'm not able to see anything properly right now. Yeah. Okay, let me go through a couple of questions again. Okay. Stick as a diffuser, centrifuge would be a nice project. Are there other advantages of using a dry mounted media compared to permanent mounted media using a mounting medium? Honestly, dry mount, let's put it this way. I'll just give you an example here. Pollen grains from flowers are very sensitive. If you use a water-based mounting medium, what they're going to do is they're going to swell up and they're going to expand in their size. But sometimes the pollen grains, when they're transported through the air, are dry. So they're a little bit more shriveled up and they shrunk together. So the question is, and if you add now some kind of a mounting medium to it, then it might also change the shape. So I would say it depends a little bit on the specimen and what you want to show. Generally, the image quality with a mounting medium is significantly better. The images appear to be brighter. And simply more clear and more contrasty. And as I showed you before, the yeast cells, they look pretty dark. However, there might be some certain cases where dry mounted slides are the way to go. For example, if you want to make dry mounted slides to be for stereo microscopy, for example, you want to put some hair on the slide and you simply want to protect it a little bit and make sure that it's flat on the slide. Because you want to observe the specimen with from the top, then this is a very easy and straightforward way of doing that. I mean, you just take a microscope slide and let's say a cover glass over here, okay, and you want to observe something. Let's say, I don't know, we can try this out right away. I don't know. Look, let's take a piece of paper, piece of tissue paper, okay, and all you want to do is you take a piece of tissue paper and you would like to see how does the dry tissue paper look like under the microscope, and of course you would like to have it completely flat. So all you do is you make I'm going to improvise this again, is just stick the cover glass using some tape. I'm not going to do the other side now, and you look at it under the microscope. Because maybe you're just interested in observing it, when it's dry. Where is this? Here it is. Okay. Why not do the following? Why not add a little bit of water? Let me see. Where is some water? I need to get myself a little bit of water. And I'm going to add some water now on the side, and let's see if this works. Just a second. A little bit of water. Yep, and this is how it looks like with water, right? And so you see a little bit quite nicely that the water, makes everything much brighter, because there is less difference in refractive index. Yeah. So this was simply a short demonstration to see how the contrast and appearance changes with air or with a mounting medium. In this case, it's water. Okay. Yeah, so people, I think, what time is it now? Oh, it's 1 hour and 24 already. Yeah, I think I'm going to slowly call it a day, or call it an evening in my case. If you have any specific comments that you would like to add, the video will be, of course, online afterwards for everyone who also joined in late a little bit. So you can also look at the beginning of this live stream. I hope that it was interesting for you again. I have to clean up the whole table and to take care a little bit of the slides that I've got here. The point of these live streams is also to motivate you a little bit to try out and to experiment and to have a little bit of fun. Because after all, microscopy is a nice hobby that many of us are actually sharing. I think next week we're going to simply have a look at how those slides are doing. No. Important announcement. I'm going to try something different also next week. I would like to do a live stream where I'm not going to show you some microscopic techniques, but where I would like to talk a little bit about biological topics. Okay, so what I'm going to do next week is it's going to be a little bit of a test run. It is in my other channel, the main Microbehunter channel. I'm going to do a live stream at the same time, but I would like to talk about a biological topic. Because over the last I received emails and there have been comments where people requested that I not only talk about microscopic techniques, but also about some scientific topics. So I'm going to try that next week as well, same time. Unless something unexpected happens, then we're going to see how that works. Okay, but it will not be in this channel because it's going to be more of a biological topic. But I hope that you are going to join in any case. Okay, thank you, Oliver. And now I'm worried about skin mites. Don't be worried. The face mites are completely harmless. Okay, so we're going to do that, so I would like to, I'm just reading the last comments here, and I'm simply going to say have a nice week, all the best, bye bye, happy microbe hunting as always, and hope to see you again. Bye bye.